Acute myologenous leukemia, also called AML. If we magnify inside the bone, we will find the bone marrow, the soft inner part of the bone where all blood cells are made. This process is called hematopoiesis. In hematopoiesis, stem cells divide into lymphoid stem cells and myeloid stem cells. Myeloid stem cells further divide into RBC, platelets, and myeloblasts. Myeloblasts are immature white blood cells which can develop into granulocytes. During differentiation, myeloblasts first differentiate into promyelocytes, so promyelocytes can further differentiate into basophils, eosinophils, and neutrophils. However, in AML, the differentiation to promyelocytes is blocked. In this way, as myelocytes cannot differentiate into granulocytes, they accumulate in the bone marrow. But why? If we dive inside chromosome 13, we will find the most common gene associated with AML, the FLT3 gene. The FLT3 gene codes for the FMS-like tyrosine kinase 3 receptor. This receptor is located in the cell membrane and activates FMS-like tyrosine kinase signals for three main proliferative pathways: STAT5, MAP kinase, and AKT. While a wild-type FMS-like tyrosine kinase 3 receptor needs a growth factor to be activated, a mutated FLT3 gene confers a mutation that causes the truncation of the tyrosine kinase receptor. Therefore, mutated tyrosine kinases can dimerize and transphosphorylate without the presence of a growth factor. This transphosphorylation activates the RAS pathway, which in turn activates MAP kinase and MYC. MYC is a transcription factor that increases cell proliferation, causing an accumulation of myeloblasts in the bone marrow. What causes AML? There are 4 to 6 different genes like FLT3 and over 50 different genetic mutations that have been associated with this cancer. Treatments for AML include chemotherapy, targeted drug therapy, and stem cell transplants. The most promising treatment being targeted drug therapy, as scientists can test leukemic cells for oncogenes like FLT3. If the gene is present, they can use specific target drugs. FLT3 inhibitors are the most common targeted treatment, which includes drugs like midostaurin, quizartinib, and gilteritinib. However, in some AML cases, the patient has a mutation in the IDH and IDH2 gene. IDH1 and IDH2 proteins are important for differentiation of myeloblasts into granulocytes. Therefore, targeted drug therapies include IDH1 inhibitors like vosatinib and IDH2 inhibitors like enasidenib. However, these treatments are usually used in people who are not responding to other therapies. In this way, AML cells can be treated with monoclonal antibodies if their myeloblasts express an AML-specific receptor called CD33. These antibodies are also called mitotarg, which binds to CD33. This binding recruits the chemo drugs so they can go inside the cell body. Once the drug is inside the leukemia cell, it is not allowed for cellular replication. This is because the chemotherapy drugs provoke so much DNA damage to cancer cells that they can no longer divide. This happens because the chemotherapy drug enters the nucleus and forms DNA adducts, so the DNA repair mechanism gets overwhelmed and apoptosis is activated, which allows for inhibition of cell division. In this way, myeloblasts can finally differentiate into granulocytes. A subtype of AML is acute promyelocytic leukemia, also called APML. This cancer is caused by a chromosomal translocation between chromosomes 15 and 17. This translocation leads to the formation of a new gene that is only found on this subtype of AML, which codes for the fusion protein PML-RAR alpha, standing for promyelocytic leukemia retinoic acid receptor alpha. In this fusion protein, the retinoic acid receptor is truncated. This blocks the promyelocytic differentiation into basophils, eosinophils, and neutrophils. APML can be treated with high doses of transretinoic acid, which helps to overcome the PML-RAR alpha actions. In this way, the promyelocytes are able to differentiate, which stops their proliferation.